All right, hello everyone. I'm Dr. Rowena Stern. I want to talk about human and marine health and what the CPR survey does, or the Continuous Plankton Recorder, long word. I work in the CPR survey department at the MBA. Just moving on to the next slide here. As the name suggests, the CPR survey records plankton groups. So that's phytoplankton, which are primarily single-celled algae, and they come in a variety of forms, you can see on the left-hand side. And it also includes zooplankton, which can be tiny animals that are either sort of permanently plankton or sort of early life stages of larger animals such as fish. But they can also consist of other things that look like animals, but they're kind of known as protists. So things like radiolaria and ciliates. They come in a variety of beautiful forms, as you can see. Additional to that, the plankton also holds bacteria and virus, of which numerically they dominate the plankton sphere. So the Continuous Plankton Recorder Survey has been going for a long time, since the 30s. So it's going to be 90 years this year, and has collected 7 million miles of plankton. The map on the left shows you primarily where the sampling has taken place. Everything in red is the CPR survey here in Plymouth, but there are sister surveys that are being carried out. There's the North Pacific CPR survey that goes from all the way from Canada to Japan, and there's several CPR surveys in the Southern Ocean, around Australia, and the Antarctic. So we really are truly global. The CPR survey, the actual device was invented by Sir Alister Hardy. It's deployed off the back of ships of opportunities. You can see in the picture on the top right-hand side, and on the bottom, it shows you that the plankton is sort of sucked in through a narrow sort of hole and captured on a piece of silk, which is sort of sandwiched between another sort of silk pieces. And it's sort of collected up on a roll, which is later cut up and analysed by scientists here at the in Plymouth at the MBA. Excuse me. And actually the analysts are incredible. They analyse over 300 types of plankton forms, and increasing number of types as the years have gone on. This is an open, on the left-hand side is this open CPR device. You can see the silk on it, which is cut up, and you can see in the middle, it's analysed by a microscope. And on the right-hand side, you can actually see what the analysts see. There's animals of different forms. Because the mesh is quite big, it mostly collects zooplankton, but because it collects so much water, it's over 3 meter cubed of water, on top of the animals, sometimes they also collect large phytoplankton and even small phytoplankton, as small as 10 microns and even less. And all of that is analysed and put into a quality assured data set, which is freely available. But additionally, and unlike many other organisations, we have a sort of a sample archive, which the samples are carefully archived since 1960, which has given us an incredible resource to go back and look, do hindcast analysis of changes in ecological distributions of animals and plants. So one of the applications of this is the development of Plankton Indicators as a measure of ocean health. And these efforts have been led by Abigail McWatters-Gollop from the University of Plymouth and David Johns. For example, there's a simplified sort of measures like the diatom versus dinoflagellate ratios and levels, and other measures, things like holoplankton, which is plankton which are small animals that remain plankton throughout their life, and mirror plankton, like this crab larvae, which will soon turn into an adult crab. And all of these creatures have effect on human health. They are food for zooplankton, and the zooplankton's food for fish, which eventually affects human, the human economy. Many people eat fish. Fisheries are very important. On the left-hand side, this paper by Bedford et al. Has combined the CPR survey data with other data sets, such as from the Environment Agency and coastal marine stations for multiple decades. And it really is quite powerful in showing the changes in plankton distributions. On the left-hand side, we can see diatoms, and it's quite a clear change. In the fact that they're increasing sort of in the North Sea and sort of decreasing around the Celtic Sea. Dinoflagellates are another form of phytoplankton, but the picture is less clear there. You see on the right-hand side, we can see the meroplankton is definitely increasing, and there are a variety of different sort of ecological reasons for that. But these types of indicators are really useful for informing people in policy and marine management, so that they can adjust things like fisheries quotas, etc., with the changing situation. Here I want to talk to you about the work that I've been doing for last year and a half, is trying to develop indicators for ocean health. I'm working with several international researchers measuring Vibrio bacteria from CPR samples. So Vibrio is a bacteria that attaches to plankton, and particularly zooplankton like copepods, and which is fed on by bivalves such as this oyster or mussels, and where it bioaccumulates to large levels. Vibrio, many Vibrios, you can see the grey Vibrio, are just harmless, and they're oceanic creatures, but some of them are pathogenic, and they can cause severe sort of gastrointestinal disease. There's a particularly harmful one that seems to be only in the tropical and subtropical regions, but more recently we're finding in more northerly areas Vibrios that cause less fatal forms of gastrointestinal disease, but it's still serious, and forms that can cause huge skin lesions, particularly affecting the immunocompromised. This is a summary diagram of the study by Luigi Vasuli in 2012, who I'm working with. And he looked at sea surface temperatures from 1961 all the way to 2004. Vibrio is a bacteria that grows, is directly related to sea surface temperatures. It'll grow more when the sea temperature rises. Around the 1980s, 1990s, there was what we call a regime shift, where there was a change in the types of plankton found in UK seas. And what he found, as you can see, there's a sort of a bar chart on the right-hand side, that the levels of total number of Vibrio cholera suddenly rose in 1998 and 2004 compared to earlier years. And he's showing and he related that increase in Vibrio to increases in sea surface temperatures. So this is an emerging threat, that Vibrio bacteria are increasing, but we don't know whether the actual pathogenic forms will also increase too, or whether they will change more rapidly. So in this study, we had a look at an outbreak of cholera in Vancouver Island in Canada, which was linked to consuming raw herring eggs, which is a food for First Nations resource. And that happened in 2018. And so we got a grant to study this. You see on the map on the right-hand side where the star is, that's where the actual outbreak took place. And the red stars show where the CPR survey is carried out. So we took samples all along the Juan de Fuca Strait, and to find out what we wanted to see is whether the type of Vibrio found in the Straits of Georgia, where the star is the same as that found in their fish eggs, because they were sequenced, and the same type that infected the humans. So what we did is we got samples, local samples from the Department of Fisheries and Oceans in Canada and from a plankton net, the zooplankton net, and we also used our own CPR samples. The middle thing is an example of sort of cell extracts from the CPR samples, which were converted into DNA. And what we did is we measured the amount of Vibrio cholera, and what we found is we could identify Vibrio cholera actually where that circle is, just on the offshore region of the Juan de Fuca Straits, which means that it's traveled all the way out and potentially moving into ocean currents. So that was very interesting. And what we did, we found it occurred, the timing of that, of where we found it was the same as the timing of the outbreak. So that was really encouraging news to show that we've identified potentially the same type. But that's not enough. We had to actually, what we'd have to do is actually identify the specific type of Vibrio cholera, because there are thousands of different types of strains. So what we've done is develop a new technique where we can see there's the tubes on the top left-hand side are DNA from all the plankton, but because they're preserved in formalin, they're not good quality. So what we've had to do is to pull out the good quality sequences that only related to Vibrio cholera. So what we did is these black lines in the middle tube are probes which bind to just the Vibrio DNA within the whole plankton DNA sample. They pull it out, and then we have what we call an enriched sample of DNA that is enriched for Vibrio cholera, and that was sequenced. We go down, you can see the pink lines are fragments of Vibrio cholera sequence, which we can match to publicly available Vibrio cholera sequences, give us an idea of the identity of the genome. And on the bottom is an example of Vibrio cholera genomes. It basically has two what we call chromosomes, large sort of sets of DNA, and the dark grey line shows where we could match the DNA with the light grey, which is the actual sort of publicly available genome. That's from an earlier study that shows that we can do that. At the moment, what we're doing is we're busy checking to see what type of strain it is, and the next stage here, excuse me, is to then see if it can if that strain is the same as the one found in the eggs and the one that made the human sick. So we're busy doing that. And it's been quite an effort to improve the DNA, but using this method, we can really now have an edge on tracking where pathogenic Vibrios go in the ocean, whether they are capable of traveling long distances and reinfecting people at the right time, so we can find out more about their ecology this way. Eventually, we can find a way to predict where pathogenic Vibrio occur, and can provide an early warning system for Vibrio cholera. So I would like to thank everyone. There was a massive number of people involved in this project. I'd like to thank everyone, including the shipping company, Matson Kodiak, Vancouver Island Health Authorities, Fisheries Canada, NERC, the UKRI, and the First Nations Health Authority, plus my partners, Dr. Martinez Ataza and Professor Luigi Vasuli. Finally, if you all want to become members, please go to this website. Thank you very much.